that's good. So those of you who's noticed on the course link, the Unit 3 summary is due on the 10th, and as you probably also realise, we're kind of slipping behind the schedule, which is unusual. You know, when you get guest speakers and we get distracted, you're bound to lose it. But we'll catch up in the end. But the significance of it is that obviously we'll probably, hopefully, touch wood, finish the Unit 3 this week. We have got a guest lecture at the end of the week, so hopefully we'll get it done in these next 2 lectures. And then we'll revise the deadline for the summary. Yeah, because the 10th is next week, isn't it? Oh, it's on Friday. It's on Friday. So, and there's Thanksgiving, I think, next Monday. To check that. So yeah, so don't worry about trying to get it finished for the 10th. I'm not sure what the deadline will be, but we'll see how we get on. And usually I'll give you 2 weeks from that. All right. So, like I say, we're going to continue. So when we left it on Friday, I think it was, we're talking about extrinsic, intrinsic factors, and we talked about pH, water activity. Then we come to one called redox potential. So going back to your biochem days, you know about redox potentials in terms of you have a reducing agent. A reducing agent adds to the electrons, whereas an oxidizing agent removes electrons, doesn't it? And we know, and the food scientists in the audience will know that oxidation is one of the key spoilage symptoms. So you'll get rancidity and things like that. But so what you try to in a food system is actually make it reducing, not only because it converts the lipids and whatnot, but also inhibits aerobic bacteria. And the aerobes are the, like Pseudomonas, they're the kind of potent spoilage microbes. So when we talk about oxidation reduction potential, we're interested in preservation, but also we'll talk about Clostridium botulinum fairly frequently. In a reducing environment or a negative redox potential, potential for Clostridium botulinum to grow. So that's why you need to know the system or know what your redox potential is. So, as I mentioned, if you've done your biochem, you'll know this upside down, round and round, and backward and forwards. So with redox potential, it's a sort of arbitrary scale, a bit like pH, it's the hydrogen scale. And as you know, it's all down to reactions, so oxidation reduction reactions and their potential. So if it's a negative potential, it's more reducing, and that will donate electrons to the more positive potential. So that's basically how it works. And you know, all reactions involve protons, and this is why it's based on the hydrogen scale, as you know. But anyway, what does it all mean to anything, to anyone? So everything's got a redox potential. So if you've got a very negative redox potential, you're more likely to be a reducing agent, you know, donating electrons, isn't it? But if you're a very positive redox potential, then you go to accept electrons. And this is why, for example, we have oxygen as the terminal electron acceptor in the electron transport chain. And one NAD or NADPH is a universal reducing agent in the cell. So we get a summation of the redox potential. As I mentioned to you, the sort of significance of redox potential is what could potentially grow in addition to food preservation and whatnot. So when we look at redox potential, the availability of oxygen is key, isn't it? Because if you've got oxygen in there, it's going to be a very positive redox potential. Therefore, you get growth of aerobes, you get oxidation of the food components. With pH, because these redox reactions basically include protons, the lower the pH, i.e. the more acidic it is, the more positive the redox potential. The ratio of reductant to oxidant, well, again, you know, basically we put reducing agents in food products and things like that. So if you've got more reducing agent than oxidizing agent, obviously it's going to be a negative redox potential. And also the poisoning capacity, i.e. the antioxidant. So if you're a food scientist, you know about antioxidants, vitamin E, and the like vitamin C, ascorbic acid. That basically buffers the change in redox potential. It keeps it negative, doesn't it? Because you don't want to oxidize the food components. And even the activity of microbes. So what you'll find if you're in a sort of a sealed pack and it's got oxygen in there, the activity of microbes will consume the oxygen, therefore converting it from a positive redox potential to a negative. And you might be thinking, well, you know, what's this got to do with anything? Well, it's quite interesting. I was involved in a sausage project 20 years ago now, and you know, this company, what was going on, they were getting sort of bloating of the sausages due to Clostridia, and they were saying, you know, we had oxygen in here just to prevent the Clostridia, and what was happening is that yeast and lactics were growing, consuming the oxygen, making it anaerobic, and then the you took off. I remember that project well. I don't know why. So, as I said, the significance of redox potential is to know what's going to grow in your product. So when you've got it exposed to oxygen, like the surface of foods, that's obviously going to be positive. In oxygen deficient environments, it's going to be negative. And with cheese, it's going to be positive on the outside, negative on the inside. Solid meat, obviously it's anaerobic, isn't it? So it's going to be a negative redox potential. And it's all got to do with what can grow. So things like Pseudomonas, which is a potent spoilage bacteria, which we'll talk about a lot in the next unit, that needs a positive redox potential, i.e. it needs aerobic environments, whereas all the pathogens, enteric pathogens, are facultative. And the reason why they're facultative is that the GI tract is anaerobic. So I remember reviewing a paper once where they're saying, oh, the oxygen in the GI tract helps this bacteria to grow, but there's no oxygen in the GI tract, essentially, because as I say, it's an anaerobic environment. So all enteric pathogens are facultative. They can have oxygen or they can't. And then you get to the obligate anaerobes like Clostridia and other sort of ones like that, which do need a negative redox potential, i.e. they die in the presence of air and oxygen. Now, the thing is, and this comes significant later on, is that oxygen is very toxic. So evolutionary wise, it was very good to have oxygen as an electron acceptor, because that means microbes could use the Krebs cycle, which basically was more efficient, using more ATP compared to just fermentation. There's always a cost for that because oxygen is very reactive. And you'll hear something sometimes about reactive oxygen species. And this is an oxidative burst. So a lot of our immune system is based on oxidative bursts. A lot of treatments or stresses we put microbes under results in oxidative bursts. And as you can imagine, with such a strong oxidizing agent there, you're going to get all damage to lipids and nucleic acids and membranes and whatnot. So cytochrome oxidase, and this is when you're doing diagnostics, you'll do an oxidase test. And what the oxidase test is doing is detecting the presence of cytochrome oxidase. But I think you all knew that. So, and we get other sort of radicals formed by other enzymes. So oxidases, or NAD oxidases, amino acid oxidases, and they produce all these radicals. And so these radicals would react with the cellular constituents, therefore causing big disruption. If they want a guard to actually protect the cell from it. And as I mentioned, the sort of radicals generated disrupt all kinds of things, from lipids to DNA to proteins. And the suicide response in bacterial or microbial cells is very well documented. This is where you get this oxidative burst because suddenly the sort of defense systems are overwhelmed in the cell, and it causes all this irreparable damage. Now, to protect itself, the cell has this glutathione system. So what happened, and not in all bacteria, Staph aureus doesn't, has an alternative system. But with the glutathione, what they're doing is it's like an electron sink. So when you generate all these radicals from the various reactions, they actually are synced into glutathione, which becomes oxidized, because you're giving electron, taking away electrons, and the radicals turn into water, and you get oxidized glutathione, which is then regenerated. But if the glutathione, oxidized glutathione accumulates, then that can be equally as damaging to the radicals. So that's the defense system. And typically when it's overrun, as I say, all the kind of cellular constituents become damaged. And in addition to the glutathione, and this happens in lactic acid bacteria a lot, is you get enzymes called superoxidase dismutase and catalase. And what they do collectively is, again, it mops up all these free radicals that would damage the cell and therefore provide protection. And it's noteworthy that all anaerobic bacteria don't have these protection systems. And that's the reason why they're so sensitive to oxygen. So if you put a Clostridium botulinum cell culture, not spores, vegetative cells, they can easily die off within about 30 seconds. Clostridium perfringens cells are a bit more robust, but even then, it doesn't take much before they're killed off. And I always say that a lot of the microbiome work that's been done, sometimes they miss this important step of keeping it maintained as anaerobic, because a lot of the cells just die off by just sampling. But yeah, that's another matter for another day, isn't it? So, as I mentioned then, glutathione is the protector along with superoxidase dismutase and catalase. And anaerobes do lack these, so therefore they're very, very sensitive. And, you know, just going back to the microbiome experiment, if you're looking for Clostridia in a sort of, say there's been an outbreak or something, you've got to take these things into consideration when sampling, when culturing. So this is why when you're culturing anaerobes, you need these special cabinets, or at least put the media with cysteine or sulfide just to be protective in terms of reducing the redox potential. So when we look at other intrinsic factors, then we look at natural antimicrobial agents, because like us, animals have to defend themselves against other microbes. You know, plants do. You know, plants are just standing there and they'll just be devoured if they weren't had a protection mechanism. And obviously microbes like to wage war, so they've got their own sort of antimicrobials as well. And these are all what we call naturally occurring, and people like naturally occurring antimicrobials because it really favors a clean label. You know, the reality is a lot of the drugs we have and lots of additives we have are actually based on in nature, because they were derived from nature. You know, back in the day when we were starting to add nitrites, they didn't have a sort of company to go to say, could I have a bottle of nitrite? They actually depended on natural plant material. And when we look at synthetic chemicals, they're obviously purified. But people don't like purified chemicals for some odd reason. They like to have this natural. You've all seen it before, and we'll talk about it as we go along. And this goes in line with clean labeling, because clean labeling, as we talked about in, I think, Unit 1, is this sort of consumer trend where they like the minimum number of ingredients, but also what they like to see are things that are familiar to them. So rather than nitrite, they'll like to see celery extract, even though they're both the same. Anyway, that's marketing for you, isn't it? All right, so a question for you then is that there's a lot of natural antimicrobials, but what do you think is most accepted by the customer? So you can click on the box, well, hopefully you can. And when we look at these different sources, like with the animals, lysozyme, we know lysozyme's in your tears. You know, that basically degrades, and it's in egg white. That's in egg white, it degrades peptidoglycan. Lactoferrin is actually found in milk, raw milk, and it's a very interesting sort of antimicrobial, because not only is it antimicrobial, but it's also a chelating agent, has DNase activity, you know, breaks down DNA. We'll talk about that. Chitosan is actually from the shells of crustacea, and not so long ago it was like a waste product, and you could, you know, basically all these wasted shells, and people would pay you to take it, and you could make chitosan and make money. But now it's such in demand. I think I heard that actually it's more valuable than the seafood itself. And there's fermentation. So chitosan's a cationic polymer like protamine. So protamine is similar. It's these cationic polymers that disrupt the membrane. And then fatty acids, there's a few of them that kind of disrupts metabolism, all kinds of things. On the microbial side, we have ruterin, which is a metabolite. It's an aldehyde produced by Lactobacillus rutei, which is a probiotic. And people think it's like an oxidizing agent, but not many people are sure. But you actually can buy it, ruterin, and it's a kind of aldehyde. I'm not sure how good it is for you, to be honest, in pure form, but people sell it. Bacteriocides we'll talk about, bacteriophages we've talked about, and plants of essential oils. Anyway, I'm rambling a bit now, I'll tell you. So we'll just see what everyone thinks, see what's acceptable. That's interesting, and he's very right. Consumers like plant-derived. They look at plants as these sort of harmless, natural sort of a source of lots of things, including antimicrobials. Obviously, people get put off animals because of this. It could be just because they're vegan, but animal-derived or microbial-derived, you can imagine they've got negative connotations. Microbes because of the pathogens, animals because of various reasons. So you're exactly right. It really is the plant which is the most popular. And plant antimicrobials, like I say, are very popular. You go to any health, we could go to health stores, which were closed down because of the pandemic, which is ironic, I suppose, in a way. What is the problem, though? Because like I mentioned to you, when you look at plants throughout history, like 10,000, 3,000 years ago, the first plant-derived medicine was described. Way before that, I'm sure it also was. But what's the problem with applying these plant antimicrobials? Is it because of the consistency, the expense, the sensory, regulatory approval, or is it some but not all of the above? So plant-derived antimicrobials, essential oils and things like that are popular, but why are companies reluctant to have them? All right. So we'll look what people think. I bet. Oh, a split decision like these. Well, most of you are right. It's some but not all. The problem with plant-derived, compared to synthetic, is the consistency. With plants, it can depend on lots of things. So how much essential oil they have. Usually, it's have they been infected by a plant pathogen, because that ups it, basically increases it. The state of harvest, the state of age. So getting consistent supply is fairly difficult, and it's expensive. For the simple reason, anything that's biological is going to be low yield, inefficient. So, and if you've gone to the health food shop and you see some of the prices they charge, you'll see why that is. But they do have a sensory impact. A lot of these essential oils, as you probably know, are to do with very aromatic compounds, and they're there not just to make sort of smells and flavors. These are the antimicrobials, because the plant uses them as a defense, to kill off moles and other plant pathogens and whatnot. Now, they don't need regulatory approval because they're categorized as something called GRAS, generally regarded as safe. And anything that's generally regarded as safe is not doesn't need what we call market approval. So when you look at regulators like the FDA and Health Canada, they don't really approve anything such. They give you what we call a no market approval required, or no pre-market assessment is required. And what that means is that, look, you can use it, but if things go wrong, don't come crying to us. It's just that we don't know any negative health things. But there are negative health things with essential oils. I remember in the early days of, what was that program? Oh, I forgot. Periscope, that's right. I don't know if you remember Periscope. It was supposed to be this sort of video, online video thing. And what people were doing, and I don't think it exists now, but it could be. But anyway, so in the early days of Periscope, people were posting their videos, like a live chat and things like that. And there was this woman in California. She was posting this chat about essential oils, and she's saying how good they are. And she was saying how she would take some neat essential oil, and it would make her healthy. She said that it really burns when you first take it, because it is. These are toxic chemicals. And when I tried to explain to her, about the dangers of having pure essential oils, she said, look, these aren't chemicals, these are medicines. So I hope she's still there. I hope she doesn't let her. But they can be, don't think you can just take them neat. There are sort of limits, and not only limits of acceptability, and limits of safety. So some, but not all of the above is correct. I think Periscope's still going. Didn't catch on though. I think TikTok's the one now, isn't it? There you go. I'm up to date. So with plant antimicrobials, then you're looking at essential oils, and the most famous ones are things like oregano, caverol, thymol. Alicillium from garlic is another, along with hop extracts. So with these antimicrobials, they're usually added to products. So the most famous one, I would imagine, is rosemary oil. And if you go to the supermarket and look at ground turkey, you'll notice nearly all of them have rosemary oil in. And this rosemary oil isn't so much an antimicrobial, although you can use it as that, but it also is an antioxidant. So all these essential oils are added, to give, and they've got to have complementary flavors. And there are sort of derivatives, as we call them, which hasn't got the flavor notes, which you can use, but they're very expensive. So, but you use them where you can use them, and sometimes they're complementary, sometimes they're not. Gram positives are much more sensitive, and because gram negatives are cytoplasmic membrane, and as I say, they've got all kinds of functions, from membrane disruption to oxidizing agents to metabolic inhibitors. So there are a wide variety of different ones. Now, raw milk is an egg, and raw eggs are a sort of source of natural antimicrobials, because in the embryo or the young baby, they need protecting against these pathogens that are going to come in and take over. And if you're in the raw milk brigade, you'll hear a lot about these natural antimicrobials, saying, when we pasteurize milk, they degrade all these natural antimicrobials, which basically makes milk unsafe, and it affects the health. Well, their argument is kind of blown out of the water a little bit, because they always talk about this lactoperoxidase system. The lactoperoxidase system is found in milk, and what it is this production of hyperthiocyanate, which is very antimicrobial. And the problem is, though, what the raw milk people don't appreciate, that you can't, lactoperoxidase system there, but you need to add substrates in order for it to work. And those substrates are thiocyanate and hydrogen peroxide. So thiocyanate are in things like spinach, green leafy vegetables. And peroxide, it can be derived microbially, but usually you have to add it. And the thing is years ago, they used to use this quite often because refrigeration wasn't available in certain parts of the world. And they claim a 5-day shelf life, which I can't believe. I don't know what the milk tastes like, though. But yeah, basically, but the lactoperoxidase system is harnessed, and you can use it in food systems. I've seen it done there. And lactoferrin is, like I say, an interesting one. It's got all these properties, from chelating agent to direct antimicrobial to DNAs or degrading nucleic acids. And they're actually using it as a wash now. So you can actually have lactoferrin washes. I'm not sure how successful they are, but it's all in sales, isn't it? And they've got other things like coagulin proteins, which basically bind to the surface of bacterial cells. And once they're bound to the surface, if they are ingested, the immune system takes them out because they're labeled with this protein. And they've got all sorts of antivirals. And, as I mentioned to you, the raw milk people really go on this natural antimicrobials, which is true. Certain pathogens like Campylobacter, very sensitive to oxidation, are quickly inhibited by these natural antimicrobials. But E. coli and Salmonella, not so much so. But they're all destroyed by pasteurization. But as I said, people look at things like raw milk and things like that as a source of these natural antimicrobials, which obviously they want. Now, a bit like eggs, though, eggs function to support an embryo, so they need to have this sort of antimicrobial to stop the pathogens killing the poor embryo. And you've got things like biotin, which sequesters iron. And as we'll talk about in the pathogen section, iron is very essential to pathogen survival. And they'll go to a lot of lengths to get iron. to the point where they'll start lysing the blood cells to get to the iron. And we'll talk about Yersinia, which is known as the vampire bacteria because of its love for iron, but we'll talk about later. Lysozyme, as you know, is in egg white. It functions to degrade the gram-positive cell wall. And now, when it comes to microbes, obviously, we've talked a lot about the organic acids and acetate and hydrogen peroxide and bacteriocins. So we'll talk about bacteriocins because we've covered the other lot. So bacteriocins, as we talked about in quorum sensing, are interesting. The peptides, and it can be different forms. There's a lot of different ranges of bacteriocins. And the thing is all bacteria produce them. So E. coli produces colicin and bacillus, pyocin, and epidermin. And what these bacteriocins do, they're quorum sensing molecules in gram-positives, but they also have antimicrobial activity, i.e. they act like antibiotics. Some bind to the cell membrane, some disrupt metabolism. Now you might be thinking, saying this is golden, isn't it? We have these antimicrobials, which essentially are natural, so we can put them on our label. But the trouble with bacteriocins, they've got such a narrow spectrum of activity. But why do you think it is? So bacteriocins are found in lots of different bacteria. You know, I would imagine, I can't think of one that hasn't got one. But they're not very useful because they've got such a narrow range of spectrum. So some only inhibit closely related strains. You do get some that do cross the general sort of barrier, but they're fairly rare. We'll talk about those. But what do you think about, why do you think most bacteria have activity over a narrow range of bacteria? Yeah, resistance is one aspect. So you basically, to be sensitive to bacteriocin, you've got to have the right receptors. You know, they don't just go in passively, but they need certain conditions to act. Yeah, you're getting close. Different cell membranes. Yeah, you need a receptor. But why do you think target-specific limited to DNA? Well, in actual fact, there's no right answer, but I've got a good theory. You're getting close. Environment fitness specific receptor. Well, what the theory goes, and like I say, no one's proven it. So the person who talked about resistance actually is very close. Oh yeah, involved resistance. So what the theory goes like is that the microbes that are going to be naturally around you are going to be very similar to you, aren't they? Because they're going to have similar characteristics to live in that environment. And so what the theory goes is that they produce these bacteriocins specifically related to the closely related bacteria that you're going to expect to encounter. And this is the reason why you get this sort of narrow host range. And you're exactly right. Like antibiotics, you can build up resistance. So the producing bacterial cell has to have its resistance or it'll be killed off itself. And obviously this passes along. So it's one of those that are philosophical, but it makes sense. Now, as I mentioned to you, there's a select group of bacteriocins that have a wide spectrum. And one is niacin. So niacin's been in the market for around about 25 years now. It's interesting. It's one of those sort of antimicrobials that you hear a lot about, but I never see it in many products, and there's reasons for that. But certainly it's used against Listeria, Staphylococcus, Clostridium, gram-positives in order to inhibit it. And it goes under different trade names, which probably disguises it a lot of the time. But certainly niacin is there. You do get resistance to niacin and things like that. So people are always on the hunt out for other bacteriocins. And as I mentioned to you, what they're looking for is this sort of wide spectrum activity. So, you know, if you go to get a culture today, you'll probably find a bacteriocin, but it's only useful, a bit like antibiotics, as I mentioned, it's only useful when it can have a wide spectrum. You know, it doesn't target a specific range of strains. So Vesicolin is from Carnobacterium vesicola, which is a fish pathogen, and oddly enough, it's found in irradiated meat for whatever reason. Pediacins from Pediococcus. So all these have a wide spectrum, and I always wonder, you know, in this sort of age of AI technology, why we couldn't artificially manufacture or synthesize a bacteriocin for our needs. But again, you know, I don't know if there's a will to do that, or even if it's possible. As I mentioned, these bacteriocins act in lots of different ways. Some, you know, bind to cell walls, so they like antibiotics, they inhibit that. Some cause pore formation, you know, to dissipate the proton motive force, and others, you know, bind to nucleic acids. And it all differs, and I say it's mainly towards gram positives, because gram negatives has this outside cytoplasmic membrane which provides protection. All right, so I think I've said this, but what limitations of using bacteria as a preservative? So with bacteriocins, then they seem to be the golden sort of bullet, or silver bullet, whichever you prefer, because they're natural. They've got this sort of potent antimicrobial activity. Is it because it's a narrow activity range? Is it regulatory approval, generation of resistant mutants, all the above, or some of the above? One more, maybe. Oh yeah, there you go. Let's have a look. Well, it's quite. Oh, that's interesting. So we've got a split decision. In actual fact, you're all right. The reason why I say everyone's right in a way is that, well, they do have a narrow range, as I kept moaning on about. Generation resistant mutants is a problem, you know, because, as I said, they need specific receptors. And if those receptors are, you know, basically modified, they get resistant. So a bit like bacteriophage, you get this natural resistance establishing. Now, where you're right and you're wrong at the same time is regulatory approval. So if, for example, because bacteriocins are from natural sources, most are generally regarded as safe, as in they don't need pre-market approval. So you don't need to go to the FDA or the CFIA and say, I want to put this bacteriocin in, can I? And they say, don't worry about it. Now, where you do need approval, though, is if you put the bacteriocins into certain products. So meat, for example, if you decided to put niacin in meat, which I don't think they do, but if you decided to, then you would need regulatory approval. And also what you need to do is if it's from a purified source, so if you're using an extract, for example, you buy these skim milk extracts. They're called skim milk powder. It's under Microguard, and I forget which company does it, but Microguard is its trade name. They're kind of an extract from a skim milk culture that's been fermented. So it's not, it's extracted, it's purified to a degree, but it's not pure bacteriocin. It's just a preparation. So you don't need approval. But if you've got a very purified bacteriocin, then you do need approval. If you've got it from a genetically modified bacteria, you need approval. So I'll give the D&E the back same sort of thing, but usually it's some of the above. Most don't need it. There you go. Live and learn. So as I mentioned, natural antimicrobials have their place, and people like them because of the sort of clean label, and consumers fall over themselves most of the time to get them. But chemical or synthetic preservatives are favoured. For the reason is that they're cheaper, consistent, and available. And you're not, with sort of plant extracts, you don't know what you're carrying over, because it's not just a purified oil, it's got lots of different constituents in. And we'll talk about each one of these in turn. We've talked about organic acids and esters before, but when we look at preservatives, most are what we call bacteriostatic or fungostatic, i.e. they're added in low concentration, you know, to prevent the growth. We don't mean to kill anything with them, but at least inhibit the growth of bacteria or even fungi like yeast and molds. But when you come to the killing, that's usually higher concentration and more effective. So when you're looking at preservatives, you've got to appreciate saying, are you inhibiting or are you killing? If you're inhibiting, then obviously you might need a kill step, a pasteurization step. But if you're killing, then that would constitute a pasteurization step, if you see what I mean. So it all depends on the type of antimicrobial and also depends on the concentration. So we'll talk about sulfur dioxide and sulfite. So in a lot of ways, if you had to pick a preservative that was most effective, like a golden bullet or silver, I think it's a gold bullet, I think it's silver bullet, a silver bullet, sulfur dioxide is it. You know, basically you can apply it. Sometimes it can leave residues if it's about neutral pH. It's an antioxidant, it's an antimicrobial, so you can see how it preserves fruit juices and things like that. And it is one of the most effective you can have, you know, in terms of ppm, only about 100 ppm, well, maybe even 50 ppm is enough to start killing things off. And it does this by acting in various ways, depending on its form. So as we'll see in the next slide, you know, sulfite, sulfur dioxide, bisulfite, all are different forms of the same sort of molecule or compound in terms of, and it depends how they elicit their sort of antimicrobial action through, you know, direct oxidation to reduction to basically just general disruption. And as I mentioned to you, whereas other antimicrobials are specific for bacteria, with sulfur dioxide and sulfites, they've got a very broad range, you know, from bacteria right up to yeast and molds. So they're very favoured. All right, so as I mentioned to you, sulfur dioxide, sulfites, and bisulfite really depends on the pH of the system. So at very acidic pHs, you have sulfur dioxide, and at alkali pH, you have sulfites. And in between those, in neutral, you have the bisulfite. Well, the question to you is, which do you think is the most antimicrobial form? Is it sulfite, bisulfite, or sulfur dioxide? And you can click on the target. All right. So sulfur dioxide, bisulfites, they're added to food systems to prevent browning. They're also there as an antimicrobial. In wine, they make it reductive, meaning that you don't get the oxidation of the wine. It keeps its colour and things like that. And it's, you know, very versatile kind of preservative. Right, I think that's everyone. Let's see what people think then. Oh, interesting. Yeah, so the most antimicrobial form is actually sulfur dioxide, which is the gaseous state. So they use sulfur dioxide for all kinds of things, like preservation of fruit juices and whatnot, and even pumped in, very toxic. So if you're in a room with sulfur dioxide, you certainly know about it. Bisulfate is the more stable one. So, for example, we're doing preventing browning. It's usually the bisulfite form. The sulfite isn't very antimicrobial. It does have an exert an effect, but typically it's the sulfur dioxide that's the most antimicrobial. Now, the problem with sulfur dioxide, though, is that it's got a very bad press. Certainly, it's not discriminatory in what it oxidizes, and so it'll take out vitamin Bs, for example, so you're not allowed to add it to meat. But it is versatile in terms of preventing browning. Now, as I mentioned to you, I think in an earlier sort of unit, they're trying to get rid of sulfur dioxide and sulfites because it's associated with these allergens. So if you want to know if you're sensitive to sulfur dioxide, all you have to do is drink some white wine. And if you get red cheeks and a red nose, you're pretty allergic to it. And you know, people suggest that it's down to all things. So what they're doing at the moment is reducing the permitted level, you know, gone from 100 down to 20. And I think there's talk in a few years to get it banned altogether. But as I say, if you ban something, you've got to have something to replace it, which they haven't quite yet achieved. So nitrates and nitrites are an interesting one. So they're used in curing agents. So if you like bacon and ham, that's the pink colour. It produces nitrosamines and or nitromyoglobin, should I say. Nitromyoglobin is a pink colour, which gives cured meat its colour. And the reason why we add it is for a few reasons. One is that it's traditional. So when you have ham or bacon, you associate it with a certain colour and taste. And that's what these nitrates and nitrates do. They make this, say, nitromyoglobin, gives it pink color, but you also get these sort of flavor profiles. So when you're doing, when you're cooking your meat on your barbecue and that, these nitrites from cured meats can produce what we call nitroamines. So these nitroamines, as you've probably read, are carcinogenic. And there was a study done by WHO, World Health Organization, about 2-3 years ago, where they came up with this headline saying, have cured meats, so like smoking 20 cigarettes a day. Totally sort of fake news, if you want to call it that, in terms of exaggerating. But with this WHO committee, what they do is essentially go around looking for carcinogenic substances. And nitroamines are when you get these nitrates reacting with proteins. But on a level, it's I think it's 1,000 times less carcinogenic than cigarettes and things like that. But who wants to swallow a good headline? But because of this concern, they've been looking for substitutes. And with these substitutes, then, is that they try to recreate it. But to know, to appreciate the substitute, you've got to know the chemistry first. So we just talked about sulfur dioxide existing in different forms depending on the pH. Well, a bit like that, nitrites, nitrates, and nitric oxide, it's not so much pH, but the degree of oxidation or reduction. So when nitrates reduced to nitrite, that's the most antimicrobial form in foods. When you go to a further reduction to nitric oxide, that's very unstable. And if you know nitric oxide, you know it's called laughing gas, isn't it? And it was reported to be a cure for COVID. So if you get virus, you can use nitric oxide. Well, I suppose you'll laugh at the prospect of it all. So nitric oxide is produced in our bodies. It's part of the immune system. It gives this very quick oxidative burst. And there is credence for that. But nitrite is what's in food in a semi-stable form. And as I mentioned to you, it does produce these nitroamines later on when it's barbecued. And if you want to prevent those, you basically dip your meat in beer, and like Guinness, because that kind of acts as a sequestering agent for the free radicals that cause chloroamines. There you go. That's a tip for the day. So nitrites have a very negative health, detriment because of the carcinogens. But nitrites are also used to control blood pressure. So typically, it's one of these things where it's bad and good. But the reality is that with nitrites, they're really effective against Clostridium botulinum and other Clostridia. And to overcome this sort of objection to the sort of synthetic chemical, what companies do is that they take nitrates from plants, and celery is very rich in nitrates, and lettuce and spinach and things like that, and they reduce it. And when they reduce it to nitrite, then they have a natural form of nitrite. So the thing is with nitrates is, and like I say, I think you know this, is that they only become a health issue really when they're basically heated in a cured meat, so it produces nitroamines. Nitrates by themselves aren't so dangerous. So we have to, as I say, consumers don't like them. They hear carcinogenic and say, God, I'm not going to eat another hot dog and things like that. So what companies have done is that they get this natural form of nitrate, reduce it to nitrite, and call it cultured celery or celery extract. So if you go to the supermarket, after you finish with the ground turkey, just go to hot dogs and see if you can see cultured celery extract or fermented celery extract. Because what that enables companies to do is say, this is no added nitrates or nitrites, but at the same time having even more nitrites, because a bit like we talked about natural antimicrobials, we've got no real control of the concentration. They're literally an extract. So in actual reality, this can contain more nitrites, but it's no added nitrites. You're not adding nitrites. You're adding, basically cultured celery extract. So it's one of those things. And I remember Maple Leaf tried to launch this, and they still do. They still got it, but they were kind of saying, "Oh, contains no nitrites." And when they got found out, Randy Hoffman, who's their chief sort of food safety person, he got inundated. I think he's still there though. He survived it. So nitrite-free foods are in demand, but what is the food safety risk? So, when all this thing about nitrates kicked off with this WHO report, this company came to me and they said, "Oh, we're very good. We've taken all the nitrites out, but what happened to that company? Did they suffer from premature spoilage? Was there a nitrogen deficiency? Was there potential for growth of Clostridium botulinum, growth of Listeria, some but not all of the above?" So, like I say, consumers and marketing people, and I see this all the time, they strive to say we need to go preservative free, we need to go additive free. And as you're a formulation scientist, microbiologist, you've got to try and give them alternatives. But sometimes people have to be safe from themselves. And that's where the excuse for nitrite say, look, we use cultured celery extract to protect the public. But what would it protect them from? Right, let's have a look. Oh, interesting, you're right. So you sum up it all. So what the company got was blown sausages for a start. The Clostridia were blowing up. You had psychotrophic bacteria there, or Clostridia, and they were basically inflating the packs. Yeah, nothing to do with nitrogen deficiency at all. Yeah, but the potential growth for Clostridia. Interestingly, Listeria is affected by nitrates, but not to the same extent. You couldn't use it as a preservative. It kind of just slows it down a bit. But the sort of gram positives of obliquous anaerobes, they're really affected by nitrates, which is why it's important to keep it in. Just have to have less hot dogs a week, I guess. So now we're going to move to extrinsic factors. Extrinsic factors are the sort of environment around the food itself. So we'll talk about temperature, gas composition, relative humidity, and resident microflora, which I always think should be an intrinsic, but it's what it is. So as we talked about in the first unit, evolution has really tailored microbes to live in specific niches. So, for example, those who lived in hot vents are thermophiles, i.e. they can exist and live under high temperatures, but you put them at room temperatures and got nothing. And a bit like other bacteria, some are psychrophiles, they only operate in the cold. Some are psychotrophic, operate in the cold and at body temperature. Whereas others like E. coli, Salmonella, they're mesophiles. They like body temperature. So, and this is all adaptation because of enzymes, isn't it? Because their sort of membranes are fluid at a certain temperature, the enzymes operate at a certain temperature. But you get different environments depending on how the microbe evolved. So they adapt by basically the composition of the membrane. So if you're a thermophile, you have a very saturated fatty acids in the membrane. But if you're a psychophile, you like cold, you would need very unsaturated fatty acids, isn't it? For the simple reason it needs to be semi-fluid, it can't be rock solid. And the enzymes, as we know, enzymes have a certain working temperature. Thermophilic enzymes are favoured for industrial microbiology applications because obviously temperature and rate and whatnot. But most of the time, the temperatures reflect the environment they're from. And when we look at the significance, obviously we're talking about culturing. We need to have the appropriate temperature, but also we need temperature to know how the microbe reacts to temperature for the simple reason we want to predict its growth. Is it going to be a danger or not? And so this is one of the important factors we have to introduce. Now, with temperature, as you probably know, or might not know, there's basically a danger zone. So basically what you have to do is have food stored cold, below 4 degrees C, or very hot, above 63 degrees C. And the danger zone, a temperature abuse for a unstable product, is between 4 and 63. Now, if you go back in history to determine why 4 degrees C was selected, it's quite interesting that it was basically just mentioned in a food safety manual from 1964, which was published by the California Public Health Department. And when they were deriving the 4 degrees C, they said, look, we have to put something down. We know refrigerators these days are around about 4 degrees C, so we'll use that. And it's quite interesting. There was a push a few years ago to have it increased, because in reality, it's 10 degrees C, which is a cutoff point really between mesophilic growth, i.e. growth for things like Staph aureus and things like that, and the other sort of psychrophiles. So, but as I say, it's interesting when you look at a lot of these food safety advices, they kind of derive from someone's best guess many years ago, and people just keep taking it forward and forward. And there's a lot of examples. I think my favourite one is the plastic cutting board versus a wooden cutting board. But I'm not going to digress. But if you look at the history of that, you know, you'll be able to see that. Anyway, I'm digressing again. So basically, we have to keep foods within this area. And there's a rule that if you're a public health inspector, you go with, in that foods can't be held for more than 2 hours at room temperature. So these are foods that are normally refrigerated. I can support the growth of microbes. And yeah, it's true that temperature abuse is the kind of thing where people store foods that should be refrigerated or kept hot. But why is that 2-hour limit imposed? So, you know, public health, if you're a public health inspector, they'll say foods should not be held at room temperature for more than 2 hours. But why is that? Is it because somebody said, well, 2 hours seems reasonable? Is it because it's the average time to travel to stores? Is it spoilage would occur if held longer? Or is it potential toxin production? It's some of the above or all of them. Oh, we're out of time again. Or just answer this question then. So, as I say, this 2-hour limit means that foods can't be held for more than 2 hours. And the reason for that is, we'll just find it. Or one more person, maybe. Yes, there you go. See what? All right. So the people who got it right, so the B, you know, basically it's the potential for toxin production. So there's two pathogens of concern. One is Staphylococcus aureus, and the other is Clostridium perfringens. And what they do is produce toxin, and the Staph aureus toxin's worse because you can't destroy it by heating. And so what they've done is they did a predictive model in terms of saying, well, you know, if a Staph aureus is kept at 20 degrees C for 2 hours, starts growing, produces toxin, and that's where it's derived from. Now, the reality is very different, but, you know, reality and fact are two totally different things most of the time. So I'm surprised no one said round number, because you could believe it, to be honest. But you're right. It is potential for toxin production. We'll talk about that in later sort of unit when we get to the pathogen one. Well, that's it for today, because we're out of time yet again, and we'll pick it up on Wednesday. Okay, you take it.